In this video, I will talk about recent strategies for getting new antibiotics. We know that antibiotic resistance is a big problem nowadays. Resistant pathogens have emerged as a consequence of inadequate use of antibiotics and insufficient infection management, which causes a danger to both public health and the global economy. You must be aware of the escape pathogens. These are responsible for many hospitalizations every year. Here, the abbreviation ESCAPE stands for 6 bacterial infections related to multidrug resistance. Enterococcus faecium, Staphylococcus aureus, Klebsiella pneumoniae, Acinetobacter baumannii, Pseudomonas aeruginosa, and Enterobacter species. And now, we have limited therapeutic options to deal with these pathogens. Therefore, people are looking for new antimicrobial compounds that can save many lives. Among the most promising new treatments for infections caused by MDR and XDR resistant bacteria include the development of novel antimicrobial peptides, AMPs, metal nanoparticles, and combination therapies. Let's first understand what is antimicrobial peptide. These peptides are small proteins with a varying number of amino acids found in animals, plants, yeast, bacteria, etc., and some are also synthesized in the laboratory. These are also called as host defense peptides. These peptides play a role as antibacterial, antifungal, antiviral, antitumor, anti-inflammatory, and immunomodulatory agent. Now, the question is, why AMPs are good future drug candidates? A recent study shows that a dose of AMP, in the micromolar range, is active enough to kill bacteria. The second advantage is, it causes rapid cell death. Third, pathogens show less resistance to this peptide. Now, let's see the mechanism of action, in brief. It causes alteration in membrane integrity via electrostatic interaction with negatively charged membranes. It inhibits DNA synthesis. Inhibition of bacterial cell wall formation is also one of the major functions of AMPs. It also inhibits the metabolic pathways by alteration of nucleic acid metabolism. The main limiting factor is that it is sensitive to proteolytic cleavage in several body fluids, particularly blood plasma, intestinal mucosa, and the gastrointestinal tract, which has a significant effect on a substance's in vivo stability. Next therapy is metal nanoparticle. MNPs alter the form and function of membranes while interfering with metabolic processes to cause changes in cell membrane permeability. Metal ions are released in the solution form, which generates reactive oxygen species and causes oxidative stress in bacteria. Metal ions may alter the structural makeup of cells and lead to abnormal enzyme activity, which disturbs regular physiological functions. It prevents DNA from unwinding and transcription. The main advantage of metal nanoparticles are, first, broad therapeutic index. Second, controlled drug release. Third, less prone to bacterial resistance. And last is, fewer side effects than conventional antibiotics. We need to study the long-term exposure of the particular metal nanoparticle, its physicochemical properties, pharmacokinetic profile, etc. The last therapy is combination therapy. For this, first let's discuss the disadvantage of single drug. Sometimes, high dose is required, which leads to toxicity. Sometimes, it causes cellular metabolism. For long-term, same, single drug administration leads to drug resistance. Now, what is the advantage of combination therapy? It shows a broad spectrum of action. Decrease the probability of resistance. It has multiple targets. It shows synergistic response. Thanks for watching.